Okay, chapter 9, population distribution and abundance. Starting to get into some of the, in my opinion, kind of the more fun ecological stuff here in the latter half of the course. Key concepts for this course are populations are groups of individuals of the same species that vary in size, both over space and time. And that species vary in their distribution and abundance across geographic ranges. Species are limited in their distribution and abundance by habitat suitability, historic factors, and dispersal. And in metapopulations, sets of spatially isolated populations are linked by dispersal. We're going to talk a little bit about this example and come back to it at the end. It's a really interesting one, although this is an image of Neurocystis, which isn't the main species that is impacted by this, but that's fine. About this, it's a pretty common story of the relationship between sea urchins and kelp. And in some areas off the Aleutian Islands off Alaska, the same is true in coastal British Columbia and off the coast around here as well. The mechanism's different because sea otters don't come into the Salish Sea. They just live in the outside of Vancouver Island, and they come down to Neah Bay, rafts of males, but they don't come into the Sound proper. But in the Aleutian Islands, there's these areas where there's really, really dense kelp forests. And there's areas where there's almost no kelp and what's called an urchin barren, which is, you can imagine just a bare rock, and they'll be literally just covered in sea urchins. Like, when I've seen them, it's, you look at an area, a meter by meter, and there'll be 15, 20 of them, and they're just sitting there. You're like, what are these things doing? Like, there's no food for them to eat. And this kind of caused researchers to have an observation. Lots of urchins, no kelp. Lots of kelp, no urchins. Urchins eat kelp. Maybe there's a relationship, but going from that observation to being able to study that more mechanistically and then use basic ecological principles to get at the larger drivers of why we see these big spatial changes has been a story that's been developing over really a number of decades, and there's actually archaeological information we can bring in about how this has happened for thousands of years. So kelps are a brown algae that form dense forest stands that harbor diverse community of organisms. And as we've talked about prior, whenever you add a three-dimensional habitat or three-dimensional habitat into a two-dimensional space, you generally increase biomass and diversity. And other islands around these dense kelp forests were surrounded by urchin barrens, which have many sea urchins and very low kelp and species diversity. It's just flat rock full of urchins. And so the grazing of kelp by sea urchins could explain these differences, but we need something, an observation is the first step into ecological inquiry, but we need a mechanism behind that. And so this is Dave Duggan's work. He was out at Friday Harbor for years. He's retired now, but a real famous old school kelp ecologist. A second, I'm trying to move this a little bit so I'm better in the window. Let's see, this doesn't want to move up. I can't move the little screen window after I start. Oh, there we go. Okay. And so being a good ecologist, one thing we like to do is exclusion experiments or removal experiments. And so in this case, they went to an area, removed a bunch of sea urchins, and then tried to see what happened. And so there was no kelp before the sea urchin removal. They went down using scuba gear, put all the urchins in bags, or this is in the 80s. They probably smashed them with rocks or hammers. But nonetheless, removed all of the urchins, and then a year later they had 20 stipes or 20 kelp per meter squared, and 2 years later over 100, about 105 kelp per meter squared, and that's a lot. So think about a meter 3 feet by 3 feet. Having 100 kelp in there is quite a lot. And so this field study was a manipulation that showed a negative correlation between urchin abundance and kelp cover. If you have more of one, you have less of the other. You have lots of kelp, you have little urchins. If you have lots of urchins, you have little kelp. That mechanism really only goes one way. The kelp aren't killing the urchin, it's the other way around. And so this was able to draw a link between urchins and kelp. But then why do we see variations in urchin? What else is controlling that? And we'll come back to that story, which it's kind of interesting. It's like one of those studies that has kind of made everybody famous in the ecological community who was involved in it. But first, let's get through some kind of the basic ecological terminology and examples we'll be looking at. So there's distribution, which is the geographic area which a species occurs. We can talk about abundance, which is the individuals within a given area. So we can define abundance as meter squared, as per hectare, or per valley. Like it's number per area. So always think of like number divided by some unit of area, something squared. And ecologists try to understand what factors determine the distribution and abundance of species. Generally, where are they? How come there are more here and not over there? Now, a population is a group of individuals of the same species that live in a particular area and interact with one another. You could think about, Lake Whatcom having a population of perch or bass. They interact with one another. They're kind of all one big population. The population size is just the number of individuals within that population, the number of bass, or say more specific, number of smallmouth bass within Lake Whatcom. And then the density is how many per unit area. So in this example, you can have a 20 hectare island with 2,500 lizards. The population is 2,500 because they're stuck on a small island, and the density is 125 per hectare. So that's just taking 2,500 and dividing that by the 20 hectares for the island. For some species, it's hard to define an individual. Aspen trees are kind of a cool example of this, where they produce via clones. They're genetically identical copies. And when a new plant grows from the root buds, where new plants grow from the root buds, and so a grove of aspens may be all one individual. And we'll talk about how we sort of can talk about clonal organisms in a bit more fine detail. And other plants and animals can form clones. Coral reefs are a big example of this as well, where an entire reef structure, an entire singular coral will be from a clone. They'll all be genetically identical. You can see it in sea anemones as well. We'll clone, they'll bud. So an example of strawberries we'll use in a little bit as well. So you can plant one strawberry and it produces those little runners that jump off. Those are all genetically identical. And so this is a grove of aspen here. And while we see, okay, well, these are all different individual trees, right? Well, they're all connected down below. And so I was trying to think of a good analogy for how this would work. Another species that's like this, because there's other, like nettle will do it, stinging nettle will do it, where they primarily produce within a patch via rhizomes. But I don't know how connected they remain. Where here the big difference is this big ball of roots is still connected. And so they're still one individual. They're all genetically identical, and the tree on the edge here will send a runner out, and then a new I keep on calling it a tree. There's got to be a different word for this in an aspen. But a new trunk will rise, and it's still connected to everybody else. It's still everybody else. It's still connected to other genetically identical aspens via this root mechanism here. So it's pretty interesting. So this could all be one individual tree, all genetically identical, and then the big differentiation here is if they're all connected via the same root network. So you can have clones that bud and then go off in two different directions. Well, those are two different individuals. They're genetically identical. But if they bud and then they just keep sharing resources and stay stuck together forever, that's kind of one individual. And so things like this make it hard to count, well, what do we mean by an individual? And it's also hard because this could have been 10 different seeds that landed and there's 10 different individuals that are intertwined, or it could just be one massive genetically singular individual, meaning they're all still connected at the root mass. We really don't know unless we dig it all up, or now use modern molecular techniques to sequence or find the genotype of all the different individuals. Bit of an aside, but the important distinction here is what's an individual? A whole bunch of what look like independent trees, but they're all connected. So they're not independent trees. They're all connected via this root mass. And so in that instance, an individual can be defined as the product of a singular fertilization. An aspen grove that would be from a single genetic individual, a genet, and here it's connected by roots, or if members of the genet are independent physiologically, each member is called a ramette, i.e. strawberries. So the big difference here is if I go down to Joe's garden and buy a strawberry plant, put it in my backyard, I bought a strawberry plant, and then it shoots a runner off, and it lands over there, and it grows a strawberry plant, and it shoots a runner off, and it grows a strawberry plant. And that little runner in between dries up and dies and breaks. Now this strawberry over here isn't getting any resources from the parent strawberry or the intermediate strawberry. They're individual members, where in the example of the aspen, they're still connected via roots. And so the genet's a bit different than the ramette. So we could think about, after splitting, what's happening here. And so this makes, there's other issues with doing direct counts of individuals. We can get an absolute population size, and so we can count with great certainty the number of cedar trees on Western's campus. They're big, they don't move, and so we can go out and count them and come up with an absolute population. And for many other organisms, it's hard. One of my fisheries professors said, counting fish is like counting trees, except for it's dark and they're moving. And so here we can use a relative population size, which we can think of in two different ways. One is the number of individuals in one place or time relative to another number. So we can estimate the number of fish in Sunset Pond by some of the methods we'll talk about, mark recapture being a primary one. And so we're using some way of estimating the population size. And then we can compare that to another area using the same method. So we did a mark recapture study in this pond. We estimated it has 500 bass. We did a mark recapture study in this other pond. We estimate it has 300 fish. So the relative population size between those two is what we'll be talking about that. We don't know the absolute number of fish in either of them, but we're employing the same methods in both locations so we can make relative comparisons. Relative size estimates are based on data that's presumed to be correlated with absolute population size. An example here is cougar tracks in a given area, or number of fish caught per unit of effort. But interpretation can be tricky. You could have one cougar running all around, making lots of tracks, or you can have 10 cougars wandering through around the same time. as well as catch per unit effort. What that means is if I go fish off the dock for an hour and I catch one fish, my catch per unit effort would be one fish per hour. The person next to me is a better fisher and he catches 3 fish per hour. So there's some differences in gear and selectivity and timing that can impact things like that as well. We can think about area-based counts, individuals in a given area or volume. Most often used to estimate the abundance of sessile organisms or plants. I'll give the example here of dandelions per meter squared, and I spelled it out to make it. That's it should be per. Not p meter. Per meter. Dandelions per meter squared. Be one thing that's, from the environmental studies building, you can look out over the Arnson lawn during dandelion growing time, and you can see all those dandelions. You can go out and do area-based counts where you throw a quadrat, count some critters, and then use that. Speaking of quadrats, quadrats are, if I was in the lab, I would bring one in. Quadrats are quad squares. Rats, I don't know what that has to do with anything, but they're typically in units of half meter squared, quarter meter squared, or a meter squared. Some are smaller, 10 centimeters by 10 centimeters. But every single one I've ever seen is made out of PVC pipe. And if you're a baller, you put a bungee cord in there, so when you break them down, they stay together. And in marine environments, we drill holes in them so they don't float away. But that's all it is. It's PVC pipe that's in a square that is some area, 25 by 25 centimeters, 50 by 50, etc. The size you use is dependent on what you're trying to measure, what you're trying to count, and how big of a sample size you need. More is better, but sometimes it's not feasible to carry around a 3 foot by 3 foot or square. And then we generally throw them out or place them in a grid. Everything we try to do in science is trying to be random. So we'll have a transect line and use a random number generator, and our transect will go from 0 to 10 meters, and the random number generator will say 5. And so we go to meter 5, put our quadrat down, count all the critters there. In my case, limpets, things like that. Here's one being used underwater, and so here's a big giant one, but they're trying to probably look at coral diversity. And so they place down this big giant quadrat, and then sit there and count all of whatever they're trying to look at within that area. And so based on that, you could say per meter squared, the abundance or diversity of corals is this, and then you extrapolate that over some other measurements you do to get an idea for the overall area of the coral reef. An example of that would be if you had this little tiny quadrat, this little 10 centimeter by 10 centimeter quadrat, tossed it around. You first toss, it landed on the ground, you counted 40 snails, you tossed it somewhere else, you counted 10, 70, 80, and 50. And so there's a great deal of variability there. And to figure out how many snails you have per meter squared, you would add up those 5 counts, divide it by 5, so now you have the average number of snails you counted per quadrat. Your quadrat is 0.1 meters squared, 10 centimeters by 10 centimeters. So you take the average number of critters you counted, divide it by the area, and in this case, you'd come up with 5,000 snails per meter squared, which is a huge number. Meters, 3 feet by 3 feet. And so if you had a 400 hectare field, that would be 2 billion snails in that example. So that's how you would go from, here it's throwing a quadrat 5 times, this little dinky quadrat 5 times, and then estimating a ginormous field out to an overall population size. Obviously, we're going to estimate something of importance by that much. We probably want to sample more, but nonetheless, that's an example. There's distance methods. which is a distance of individuals from a line converted to estimates of abundance. This is used in dendro work, tree work, a lot, where you've got a line and then the thing you're trying to count isn't super abundant, and so you put a line down the middle of a forest stand and then measure out to see how far the nearest tree is. Line transects are something we do a fair amount, where we lay a line down and then count all of the critters within a meter of that line. And so instead of a quadrat, which are these discrete sampling units, you can imagine you're sampling like a giant rectangle. So it could be 10 meters long by a meter on each side, and you just go and count everything in that giant area. And then you can use detection functions that convert distance measurements to an estimate of absolute population size. So here's a line transect where there's the line down the middle. We have distance 1 to this tree, distance 2 to this tree, and distance 3 to this tree. And then you can figure out what's the overall density of trees based on that. Oops. Mark recapture. We actually did this with my girls in the kind of early pandemic times for homeschool. It was in the spring, and there's tons of, I looked them up. It was like a yellow whirl snail. I forget what it is. It's whatever the common garden snail we have here is. And they're all over in our yard, and so we went out one day, captured a bunch of them, brought them in, and then measured them and painted numbers on them. And we weren't doing a proper mark recapture. I was actually interested in distance, how far they can go and how quickly. So we put them all out in a known location in the yard and put a stake there. And then the next day went and found them again and measured the distance from that stake in the compass direction. But nonetheless, that's effectively a mark recapture study, where you go out, you tag a bunch of individuals, you release them, you come back, capture a whole bunch of the same species, and then look at the ratio of the marked to unmarked members of the population. So if you have a tiny little koi pond in your backyard and you catch 5 koi, tag it, put it back out, go out the next day, catch 6 koi, 3 of them are tagged and 3 of them are not, you would estimate your total koi population at 10. We'll go through that calculation. But the idea is that there's a ratio of how many you caught that are tagged versus untagged. So this is used a fair amount in fish. What else uses mark recapture? I'm not a wildlife person, but they do with birds. They do more, particularly waterfowl. They'll do a lot of tagging, but a lot of that's around migration as well, is a big question. But tagging, you could do for things like salmon in the lower parts of the stream. You could tag them, and then as they go upstream, sample again to see what percentage you caught. So in this example, we captured 23 butterflies and marked them. Let them go later, the next day likely. We captured 15 butterflies. So the M unit is the number marked. The C unit is the number captured. 4 of which were recaptured. That's the R. So we can use this information to estimate total population size. This case is N, which is a pretty common unit for population. Well, N like number. I'm way low here, sorry. And so to calculate the N based on that, it's the number marked times the number captured divided by the number recaptured. So in this case, we had 23 butterflies marked, 15 captured the second time we went out. 4 of those were recaptured. So we have a population of 86 butterflies. Dispersal is the movement of individuals. And when we're talking about dispersal, we're looking at a population. So we're going to ask the question, how does dispersal impact this population? How does dispersal impact the population of snails in my front yard? Well, there's some that immigrate in from my neighbor's yard because they like my grass better. And there's some that I always have a hard time with the opposite of immigrate and then emigrate out. So you have immigration and immigration. So I can't pronounce it differently. Immigration and emigration. Some that leave the population. And so if those are in balance and all else is equal, presumably your population is stable. If the grass in my yard is much tastier, then through immigration, I'll get all of my neighbor's snails if they can somehow sense that my grass is better. The metapopulation is a group of geographically isolated populations linked by dispersal. So meaning that, trying to think of a good example for this one. Squirrels doesn't really work. Maybe squirrels in parks would work, where you've got a park with a fair amount of squirrels in it, and then there's other pockets, like an urbanized environment, there's other pockets that are still linked. So there's some dispersal between them. Metapopulation becomes really important in genetics because you'll have populations where you have two different populations, maybe they're each 1,000, and you don't have 100 or 200 going each different direction, but you have a few going in either direction every generation, which is enough to keep the genetics homogeneous. It's enough to wash away any genetic differences that are forming between the two populations. And metapopulations, we'll talk about this a bit more, are important because they can recolonize areas that are lost due to episodic events. But generally, we're looking at the population level for study, and then the metapopulation is the overall, you can think of it as like a network of populations, like a web of populations. And the geographic range or distribution of the species might consist of multiple metapopulations. So if we're looking at, this is where, like, I'm a marine ecologist. We are looking at population of amphibians that live all throughout Washington state. Likely these amphibians are not moving from western Washington to eastern Washington. There's a big barrier there. There's a big divide. But when they're looking at the, I'm going to make up a frog, the blue-bellied toad, that, if we're looking at the distribution of the species, we count western and eastern Washington, the entire existence where this animal is. But if the western Washington population went away, these frogs aren't going to crawl across I-90 and make it over to the west side of the state. So the west side of the state could be considered a metapopulation, that there's some genetic exchange and individuals that move up and down, but they're not jumping across the Cascades. And so you can have a geographic range of what I call them blue-bellied frogs or blue-bellied toads that goes across all of Washington, and then multiple metapopulations within that, an eastern Washington metapopulation and a western Washington metapopulation that are separated by a geographical barrier. So this is an example of that's a plant, it's a herbaceous perennial plant that exists here. It's in the geographical range. It's Nebraska, Kansas, and Missouri. And then there's a group of populations, a metapopulation within a certain part of Missouri. Within that, there's individual populations. And so this is kind of a heat map. Darker is more densely populated. So these plants like this area and this area, not so much in between. This is 100 meters, and so this is a relatively small area. So if they live in a certain type of habitat, in this case limestone meadows, and so it's not like they could live anywhere. They live in a very specific habitat that's really driven by geology. And so you'll have a population in a drainage, and then the next drainage over, there'll be another population. And so that can form a meta-population. But the whole overall geographic range would be, in this case, a tri-state area. Dispersion is the spatial arrangement of individuals in a population. We'll look at 3 different examples. So this is how individuals are organized. There's regular dispersion. Individuals are evenly spaced throughout their habitat. Random dispersion, individuals are spaced randomly, or clumped dispersion. Now, one example of regular dispersion we can see, and it's, I'm trying to think of there's probably a good plant one. I don't have one, but we can see a semi-regular dispersion of particularly organisms that have some sort of defense against other members of their species. And so there's some burrowing, there's some worms, some that come out and they deposit feed. So they have like a little patch of the mud. Sorry, these worms are marine worms, annelids, or polychaetes that come out, and then they feed in this little area, and then there will not be another individual in that area because they'll fight. So there'll be another one just over, and so you'll have all of these little circles regularly dispersed along the bottom of the ocean, because if they're too close to one another, they'll fight and somebody will win and somebody will lose. But they're pretty much covering the entire bottom. And so it's like these like very evenly spaced polka dots across the bottom of the ocean. Random dispersion, you could think of that as like dandelions. They just show up somewhere and they're kind of like randomly all over the place because that's where the seeds happen to land. And clump dispersion is pretty common, which I think of stinging nettle that way, although it does follow along dispersion or disturbance gradients. But generally, there'll be a clump of something like that because the habitat's just right. In that case, the way it reproduces is generally via spreading versus by seeds. So you have these really clumpy patterns, so they're based on like micro niche habitat. There's just enough sun, the soil is just right enough that it can take over and dominate in these little tiny patches. And so generally when there's like really a specialized need, you kind of have these clumpy distributions. And part of this is dependent on spatial scale as well. So in our deposit feeding polychaete example on the bottom of the ocean, it would look kind of like this. You'd have a little polychaete here, 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 here. They don't get too close because they'll fight, and pretty much every piece of the bottom of the ocean's covered, and there's not a lot of habitat variability, right? It's just like miles of mud that's more or less similar, so there's not a lot of microhabitat niches that they're getting on. Random distribution. This looks like dandelions on my front lawn. They just show up where they show up, and you're like, I don't know. I don't know why you're all over there, not over here. And then clumped, again, this is, you can think about this as a forest, and these are your stinging nettle patches, which are open to some light, and they're, kind of moist enough for them, or have whatever they happen to need. And so you have these little clumps. I'm a bad botanist, but plants exist in clumps to some extent. You can have like a big patch of ferns would be an example. Stand of cedars would be another example of that. Geographic ranges can vary. Little known fact, the devil's hole pupfish is my favorite organism. The devil's hole pupfish, I've actually never seen the devil's hole pupfish, but I've seen other pupfish. The devil's hole pupfish lives in a hole that's 7 by 3 meters across. And these pupfish, it's basically stuck there since the last glaciation. But these pupfish usually live in these aquifer holes, and then during rainy seasons, before the water table is drawn down so much, those aquifer, there's just literally holes in the ground with water. And this is in Nevada and other areas of the Southwest. You'll see the same thing. The ones I've seen are Death Valley has some in Arizona. I think those are the two main areas. But when it rains and these aquifers connect and form like ephemeral streams, and pupfish can move back and forth. Interesting about the Devil's Hole pupfish is the water table it lives in, the groundwater it lives in, it probably was and probably still is one of the main sources of water for Las Vegas. And so Las Vegas this is in the 70s, I think. Yeah. Las Vegas was pulling a ton of water as the whole casino industry was starting to build out there. And they're drawing down the water table, and the Devil's Hole pupfish was going to go extinct because of this. And so there was a big movement to save the Devil's Hole pupfish, and bumper stickers and protests, and the whole area is fenced off now. And because they had this one little rock that they would breed on, and that's the only place they would breed, and it was getting ready to be exposed to the air. The water was going to follow below, I call it their love rock, but fall below their breeding rock. And that was going to be the end of the Devil's Hole pupfish. And so because of this, Las Vegas had to dramatically change its water use and development. So this little tiny fish that nobody knows about, that lives in this little this little tiny hole, in Las Vegas, stopped, Sin City from growing and really made them figure out alternative pathways for water and development. I just think it's pretty interesting. There's 100 of them, maybe. Maybe. There's no ecological value to them. I mean, they're a critter that lives in a hole. There's other pupfish out there. If those ones went extinct, it would not be the end of all pupfish, but it would be the end of that genetic line of pupfish. And so, I think it's kind of it's a total aside, but it's a pretty interesting story. Many tropical plants have small ranges. In the late 70s, they found 90 new species on a single mountain range. And so particularly things like that are, one, plants, but then two, they can't really move anywhere. They're in these little niches that are just right, and there's desert probably all around them, and they're in a small rainy area. They just really can't move, and so there could be vestiges of prior climates that have just been hanging on for thousands of years, and that's the only spot they're found now, because 100,000 years ago the climate was much different and they were more broadly distributed. Now there's these few little pockets that are just kind of hanging on. And an endemic species occurs in only one location and nowhere else on Earth, and so Devil's Hole pupfish would be an endemic species to that area. And then you can have other widespread species. A book I've been wanting to read is, I think it's called American Coyote. But it follows the history of coyote. And I believe they're now more widely distributed than, or of higher populations now, than at the time of European arrival to the Americas. But they're very adaptable. They live well in urban areas, missing small dog and cat posters, likely coyote. And then only humans, Norway, rats, and E. coli live all over the continent. There's got to be another bacteria that lives all over the continent. E. coli is a gut bacteria. So it's like wherever we are, they would be there too, but they're also in other warm-blooded organisms. They're ubiquitous. E. coliform is the main, E. coli is the main bacteria in fecal coliform. It's the fecal coliform indicator bacteria. So there's got to be some other bacteria that are everywhere too. Geographical range includes all areas of the species that a species occupies during all stages of its life. And so this is important. The monarch butterfly is a good example where larval stages are in one area, adult stages are another. Migratory waterfowl, the same thing. Eggs will be laid, birds will be hatched, and then they'll fledge and then fly thousands of miles away. So the geographical range of the species is quite large, but where they're occupying at any certain time might not be that large. And so you can think about all the birds that go up to Alaska or up north in the summer and then south in the winter. So their range is huge, from Mexico to Alaska, but they're not occupying that whole range at any given time. They're only there during certain seasons. But if their habitat is lost at either end of those spectrums or along the flyway, then the whole population dies. They need all of that intact. This is a figure of kangaroo rats. Sorry, I'm thinking of chapter 5 or 6 of kangaroos, real kangaroos this time. Kangaroo rats are cool, by the way. I've held those. I've never held a kangaroo. A kangaroo, pictured here, have different densities in Australia. It turns out Australia is not just all kangaroos. But certain areas, there's lots of kangaroos, and it's caused issues with ag, and there's some human interactions there with red kangaroos. And so their geographic range is most of the continent of Australia, but they have various abundance throughout that geographic range. So this is looking at Dorset in England, where area of the heathlands. Yeah, so this is just looking at patchiness. So an area where, the Dorset heathlands, which is this plant here, but looking at the patchiness here. Sorry, I'm getting all sideways here. It used to be spread along this 1759 area. Then as developments happen, you start getting patches, and this is typically what happens when we have development. It starts to increase patches where organisms aren't able to move back and forth. They're isolated from dispersal, and so where before you could have dispersal happening throughout this entire area, now you have a patch here, a patch here, a patch here, a patch here, and they're not linked by dispersal, meaning if this patch goes away, individuals from this patch are not going to recolonize that area. And so this is one thing we start to see as we have development, that we're breaking a lot of what in wildlife they call like wildlife corridors, how animals move from one spot to another either as part of seasonal migration or as part of dispersal. And so as we increase the fragmentation of populations, they get smaller, they're less connected, and they're more likely to crash or not go extinct, but to no longer inhabit that individual patch. And so species distribution models can predict the geographic range. If the range is unknown or predicted to spread, invasives, etc., or shift in range due to climate change. And so we can look at this as similar to the habitat models, where we can put in suitability indices, type of cover, vegetation, temperature, precipitation, and say, okay, where should this species be based on our distribution model? And so here they took Madagascar and put it in these 1 by 1 kilometer grid cells, and then presumably using satellite data looked at things like vegetation cover, vegetation type, precipitation, temperature, etc., and then predicted where 11 different chameleon species lived, and they were right about 75 to 85% of the time, which is pretty good. And then those other areas you can look at is, well, where is that marginal based on the model, or are there other factors that are in play here? One thing these models might not take into consideration is either A, competition, or B, other excuse me, biological interactions, i.e., this area is a suitable habitat, but there's something there that eats me, and so whenever I go over there, I get killed. So it's a good way of understanding those habitats, and then when there's a disconnect between what looks like good habitat and the existence of that organism, then we can start to investigate why that might be. So habitat suitability is looking at the abiotic features: moisture, temperature, pH, sunlight, nutrients. Some species can tolerate broad ranges, generalist of physical conditions, and others have more narrow range. They could be specialist. It depends on the species. This is an interesting example of the creosote bush and the saguaro, and their differences in range, where the creosote bush is very tolerant of dry conditions as well as cold, so it has a much larger distribution, where the saguaro can tolerate dry conditions. It's just the emblem mascot maybe for Taco Time. It's that kind of famous cactus with, like you think of in Wile E. Coyote type cartoons with the big upturned arms. You can swell and then shrink with water, so it can tolerate dry conditions quite well. Creosote bushes are pretty cool as well. But the saguaro has a much smaller range because it's temperature limited in relation to cold temperature. So the desert's hot, but it also gets quite cold. And so it's the cold temperatures that are preventing saguaro from taking over a larger area, not the warmer temperatures. Okay, I'm going to stop there, and we'll knock out another one or two here. We'll see how it goes.